Thank you, George. Let me share my screen. So here is actually the contents of my talk. It will be divided in 3 sections. The first section will be basic concepts about the surfer technology and SSM-based electrophysiology in general. I will talk about advantages and also limitations of that technique. Then I will switch to the TMEM175 data we collected on different surfer instruments. And in the last 2 slides, I will highlight some results that indicate that the target membrane, where TMEM175 sits in, actually affects the compound pharmacology that is found. So starting with the basic concepts. As George already introduced, the surfer instruments are essentially pipetting robots. They will inject solutions containing the substrate of the target protein, thereby introducing a substrate gradient that acts as a driving force for the transporter or the channel, and at the same time as a stimulus for the recording. So once you apply a substrate, the substrate gradient will trigger the flux of charge across the sample membrane. And then the sample membrane will be charged. There will be a voltage generation. And that voltage generation acts as a counterforce for the substrate gradient, which is applied. And thereby the current you will record will decay within a few hundred milliseconds. And what we typically use as a readout is the peak, where no significant voltage was generated due to transport. So that will essentially reflect the steady state transport rate under the given conditions, which means the substrate gradient you applied. So let's have a look here at the list on the left. This will sum up some key features. First of all, the surfer or SSM-based electrophysiology in general will allow for real-time recording. So you will observe time-resolved currents. And from these currents, you can even derive kinetic rate constants if you are measuring not only transport, but some other conformational transitions within the protein that are triggered by the substrate binding. The surfer is HDS compatible, as George already mentioned. We have the surfer 96 SE, which is able to measure 96 sensors at the same time. A very big and interesting aspect is that you can access any kind of membrane. You usually purify membranes of a specific source. You can purify organelles and attach it to the sensor. You can purify protein, reconstitute them into proteoliposome and attach them to the sensor. We even did some recordings on whole cells, so there is essentially no big limitation regarding the source. Then you have a very high sensitivity. This comes from the pretty big sensor surface of about 3 millimeter in diameter, which allows about 1,000 times signal amplification compared to patch clamp, which is limited by the surface of the living cell. And this allows to record also slow transporters, which are hardly accessible via conventional electrophysiology. And the sensitivity is even so high that you can measure conformational transitions within a transporter triggered by substrate binding. And last but not least, talking about reliability, success rates are pretty high. You are not limited by gigaseals or the variability of living cells. So actually, so average success rate, I think, is 98%. You can do sequential measurements using the same sensor. because the attachment is pretty stable. So you can perform like 20 measurements in a row, and you will get very reproducible currents over that time period. And last but not least, the sensor-to-sensor variation is also quite low, so you can compare currents also across sensors. Coming to the key limitations, and there are two pretty big ones. The first one is that there is no direct voltage control, and that sets the technology really as a complementary one to conventional electrophysiology. That's just because you essentially measure the change in voltage once you apply the substrate gradient across the membrane. So if you would clamp the voltage, there will be just a baseline. And then as an advantage, there are no leak currents, essentially. But on the other hand, instead of leak currents, you will observe artifact currents coming from ion membrane interactions. So if you perform a sodium concentration jump or potassium concentration jump, these will interact with the lipid head groups that are partly negatively charged. And then you will get some capacitive currents as well. And that's why you need negative controls to be performed. Negative controls don't have the transporter of interest, and you only see the membrane ion interaction, and you can subtract these currents from your sample currents, essentially. Here is, again, a slide showing how a measurement trace looks like. George already showed it just briefly again. So you essentially need two solutions at least. One non-activating solution, you record the baseline, then you switch to the activating solution, which contains the substrate of your transporter. You measure a peak current because the initial transport rate is highest once you apply the full gradient. Then actually the chemical gradient decreases over time while the potential across the sample membrane increases, and that will lead to a current decay. Then you wash off the substrate again, you measure the efflux, and well, the whole thing takes 3 seconds. After that, the initial conditions are restored, and you can measure with a different condition, with different substrate concentration, applying inhibitors, enhancers, and so forth. Here are the two devices we have. This is the Surfer N1, a single well device where you can record up to 150 data points per day. And then we have the 96 well plate based device, the Surfer 96 SE, where you can record up to 10,000 data points per day. Of course, you have, given the different hardware, also differences in the liquid handling and in the software. In general, the Surfer N1 is an easy to learn instrument. It is very flexible for assay development, while the Surfer 96 SE comes also with a very powerful automated data analysis software, and is very suitable for secondary screening, for example. So this was about the background about the technology. Now we are coming to our results we got from TMEM175. We actually applied both Surfer instruments. I will note which data sets are from which instrument. Here on the left-hand side, you see full traces now recorded on the Surfer N1. In blue, it's TMEM175, and in red, it's control lysosomes without overexpressing TMEM175. Our standard conditions are essentially a 140 millimolar sodium chloride background, and then we switch from, we remove 50 millimolar sodium chloride and replace it by 50 millimolar potassium chloride in the activating solution. So we essentially see potassium flux in this trace here, and potassium is provided here at 1.1 seconds. So you see about 12 nanoamps potassium flux signal, while you see -1 nanoamp in the controls. And what we actually used are purified lysosomes for both sample and control. So we have HEC293 cell lines, overexpressing and not overexpressing TMEM175, and we purified lysosomes from this cell line via a sucrose gradient ultracentrifugation. So the first thing we did is looking at the ion selectivity, and we did this by performing 50 millimolar of cation concentration jumps against 50 millimolar NMDG. So essentially in the non-activating solution, we had 50 millimolar NMDG, and in the activating solution, we have 50 millimolar of the different cations listed here. And we did the same experiment on the control and on the sample. And here the bars actually reflect the peak currents subtracted. So essentially TMEM175 peak current minus control peak current. And from the currents, you can see that TMEM in our assay conducts potassium, rubidium, and cesium, but not really lithium, sodium, and choline. We also tested 4AP. So 4AP is an inhibitor, a known inhibitor pore blocker for TMEM175. And we repeated the measurements, both again, control and TMEM sample, in presence of 4AP, and the corrected signal is here in orange, and you can see that all fluxes, potassium, rubidium, and cesium are fully blocked in presence of 4AP. We then wanted to look into the concentration dependence for the potassium flux, and here we provided concentration jumps from 1 millimolar to 300 millimolar. This experiment was actually carried out with the Surfer 96 SE. So we used one column of sensors, so 8 sensors for each concentrations. We performed the experiments on the sample again and on the control. Again, for the control, you see negative currents, while on the sample, you see positive currents reflecting the potassium flux. And when you subtract the currents you observe with the control from the TMEM currents, you get a curve that looks like here. It looks kind of hyperbolic, but in fact, if you look at the first few data points at low potassium concentrations and the last 4 data points at higher potassium concentrations, it's very nicely fittable using a linear regression. And from the linear regression, you can actually achieve the permeability for potassium, which seems to be concentration dependent. So we keep that in mind. There are 2 permeabilities for potassium depending on the potassium concentration range. We then wanted to know something about the pH dependence. As you all know, TMEM175 not only conducts potassium, rubidium, and cesium, but also protons. We started out to investigate the effect of pH on the potassium flux through TMEM. In the upper figure here, you see the typical measurement of potassium flux, so 50 millimolar sodium against 50 millimolar potassium jumps as a driving force. And we did that experiment using different pH values from 3 to 10. And as you can see, there is a pH optimum close to 8. And then you see downregulation in the acidic pH range and in the alkaline pH range. The acidic pH range, actually, the downregulation is described by 2 different pK values. So you see like 60–70% activity loss with a pK of 7. And then again, the rest of the activity is lost with a pK of 4.5. These pH experiments were performed using the same pH values inside the lysosomes and outside the lysosomes. So in the next step, we wanted to figure out where this downregulation comes from. Is it the internal or the external pH? And therefore, we carried out a similar experiment, but we kept the external pH to 7.5, and we only changed the internal pH. And here you can see the curve that comes out from this type of experiment. And as you can see, if we acidify the lysosomal pH, actually the signal increases, the activity of TMEM175 increases, which makes sense, right? Because TMEM175 is a lysosomal cation channel, and it has naturally acidic pH environment from the internal side. But if you go lower in pH, you see the signal loss again. We think this signal loss is the same signal loss as we see at symmetrical pH, just due to competition with protons. So since you have much more protons at acidic pH values, protons go through the channel as well, and that will block the potassium flux, which we measure here. So as a conclusion, this downregulation we measured at symmetrical pH values must be due to changes in the cytosolic pH environment. And we think this is a kind of a mechanism to stop further acidification of the cytosol once the cytosol tends to acidify. So this was the effect of pH on potassium flux. Now we wanted to measure the proton flux directly through TMEM175. And this is done by pH jumps. So we did two different types of experiments. The first experiment was equilibrating the pH to 7.6 and then acidifying from the external side, essentially measuring proton influx driven by the pH gradient alone. And you can see here, these are the currents we measure with the different delta pHs. Of course, the higher the pH gradient, the bigger the current. And with the highest pH gradient, 3.0, which is the orange curve here, we also added 10 millimolar 4AP to block proton flux through TMEM, and we get this black current here. And what is remaining here is the artifact resulting from proton-lipid interaction. We actually measure exactly the same current when we measure control lysosomes without 4AP. So that's all nice. We actually measured the control as well and subtracted the currents. And here are the artifact-corrected currents and plotted against the proton concentration. Instead of delta pH, it's delta proton concentration. And here, if we fit linearly, we can get permeability for the protons. Here you can think of why is this here not linearly increasing at higher proton concentration jump. And the answer is that you actually see downregulation at acidic pH values. So if you acidify much more beyond neutral conditions, then you see that downregulation effect, and you actually don't see a linear increase. So we just used the first few data points to derive the permeability. We did the same thing with efflux. So we have as initial conditions the pH equilibrated to 4.6, so very acidic, actually resembling natural lysosomal pH. And then we provided more alkaline pH values from the external side and measure proton efflux. And here essentially you see the same thing, only if you jump to very alkaline or close to neutral pH in the outside, then you will see a linear dependence, and you can get the permeability for protons, which is essentially the same as for influx. So there is no different permeability for influx and efflux in our assays. And one very nice result is that if you take the ratio of the permeability for protons and the permeability for potassium, you will get a number which is 48,000, meaning proton permeability is 48,000 higher compared to potassium permeability. And this matches very nicely to the permeability ratio found by Hu et al. He applied patch clamp to obtain this ratio, and this validates the surfer or SSM-based electrophysiology approach very nicely. We then also measured some compounds, some tool compounds. I will just go very briefly through that. A typical workflow sequence here is that we do 3 times activation of TMEM175 using 50 millimolar potassium. This is essentially the reference current we have. And then we incubate, we provide the compound, we incubate for 5 to 15 minutes, depending what kind of compound we have. You need larger incubation time if compounds are hydrophobic and need to integrate into the membrane, so that takes a little more time. And then we repeat the activation using 50 millimolar potassium in presence of compound. Here you can see the result for an enhancer, and here the DMSO control. And after that, you can wash out the compound again. And you can see that for some cases, in some cases, for some compounds, you can wash it off. For some others, you can't wash it off. If they are very sticky, hydrophobic, they just stay in the membrane and you won't see the washout. We actually measured inhibitors, zinc and 4AP, and derived IC50 values. And we also measured enhancers, DCPIB on the left and arachidonic acid on the right. So this is the last section of my presentation. And here I actually only want to provide a kind of teaser. It seems, or some results we found indicate that the target membrane is very essential and affects the compound pharmacology when assaying TMEM175. So first of all, I would like to go back to the assay conditions we have with the surfer instruments. I told you we use purified lysosomes, but that's not the full story. So yes, we purified lysosomes. We use 2 different techniques that have been published by Schulz et al. And by Jin et al. 2008 and 2017. But we also checked for lysosomal and plasma membrane markers of these purified lysosomal samples. And as you can see here, with both techniques, we get LAMP1 concentration, which is the lysosomal marker. So there are a decent amount of lysosomes there. But we also find sodium-potassium ATPase, which means there is some contamination with plasma membrane vesicles. So essentially, since for the surfer we measure macroscopic currents, we will have or likely have a mixture of lysosomes or TMEM characteristic in lysosomes and TMEM characteristic in plasma membrane vesicles. Why do I tell you that? We did a study together with Cerevel as a collaborator, looking at 8 enhancers, potential enhancers for TMEM175. And we performed surfer assays, as I just introduced, but we also performed whole cell automated patch clamp using the SynchroPatch, where we actually have only TMEM in plasma membrane. And we also performed manual lysosomal patch clamp, where we have 100% of the TMEM residing in the lysosome. And Interestingly, the enhancers we tested can be clustered into two groups. Cluster 1 has the enhancers in cluster 1 show very similar potency with all three techniques. In the cluster 2, lysosomal patch clamp shows no activity, while whole cell automated patch clamp shows quite large activity, and the surfer assay actually is somewhat in between. And that's why I was mentioning that we have here a mixture of plasma membrane vesicles and lysosomes. So that could explain why the surfer is basically in the middle of these techniques. And for cluster 3, it's a very similar thing, but here we see high activity in lysosomal patch clamp, essentially no to low activity in whole cell patch clamp, and the surfer again is somewhat in between. Why can that be possible? So our first thought was, well, if a protein resides in a membrane and interacts with lipids there that are only available in that target membrane, or also with other proteins that are only available in that target protein, the interaction sites of the compounds will change, right? So that could have a major effect. And luckily, just a few weeks ago, there was a publication showing that the LAMP proteins actually directly interact with TMEM175, and that could explain these changes. So we think that it's really important to look for compound potencies of TMEM175 in its natural environment, which will be the lysosomal environment. So that was it from my side. I just wanted to highlight a few resources. There are two nice book chapters from 2017 and 2020 where you can dig into the surfer technology in general, and also just released this year a publication which highlights more the data analysis part, also looking into pre-steady state currents, binding currents, and transport currents, and how to distinguish those. And the TMEM175 story is actually currently under review, so we hope that this will be released soon. And if you want to see the Surfer N1 in action, there is a YouTube video you can access here, where Maria will show you sensor preparation and guide you through some measurements. And now I would like to hand over to Adriana, who will continue with some results on TMEM175 on both SynchroPatch and Surfer.